Major depressive disorder, MDD, is a common and debilitating mental health condition that affects millions of people worldwide. MDD is characterized by persistent and pervasive feelings of sadness, hopelessness, guilt, and loss of interest in daily activities, as well as physical symptoms such as fatigue, insomnia, appetite changes, and pain. MDD can impair one's functioning in various domains of life, such as work, school, relationships, and health. MDD also increases the risk of suicide and other medical complications. The causes of MDD are complex and multifactorial, involving interactions between genetic and environmental factors. In this article, we will explore how genetic predisposition, environmental factors, and epigenetics may contribute to the development of MDD. Genetic factors and their role in MDD. Genetic factors refer to the variations in one's DNA that influence the risk of developing a certain trait or disease. Genetic factors can be inherited from one's parents or arise from spontaneous mutations during development. Genetic factors can affect the structure and function of the brain, the production and regulation of neurotransmitters, chemical messengers that mediate communication between brain cells, the response to stress and other stimuli, and the susceptibility to environmental influences. Several lines of evidence suggest that genetic factors play a role in MDD. First, family studies have shown that MDD tends to run in families, meaning that people who have a first-degree relative, such as a parent or sibling, with MDD have a higher risk of developing the disorder than those who do not. Second, twin studies have estimated that the heritability of MDD, the proportion of variation in MDD risk that is attributable to genetic factors, is about 40%, which is comparable to other complex diseases such as diabetes and hypertension. Third, genome-wide association studies, GWAS, have identified hundreds of genetic variants that are associated with MDD risk at a population level. These variants are mostly located in genes that are involved in various biological pathways related to MDD, such as neurotransmission, stress response, neurodevelopment, neuroplasticity, inflammation, and circadian rhythm. However, it is important to note that genetic factors alone cannot fully explain the occurrence of MDD. No single gene or variant can cause or predict MDD by itself. Rather, each genetic factor has a small effect on MDD risk and interacts with other genetic and environmental factors in a complex and dynamic manner. Moreover, genetic factors may vary across different populations, subtypes, and stages of MDD. Therefore, more research is needed to elucidate the specific mechanisms by which genetic factors influence MDD. Environmental factors' role in major depressive disorder. Environmental factors refer to the external conditions and events that affect one's development and well-being. Environmental factors can be physical, such as temperature, light, noise; chemical, such as drugs, toxins; biological, such as infections; psychological, such as stress, trauma; or social, such as relationships, culture. Environmental factors can have positive or negative effects on one's mental health depending on their nature, intensity, duration, timing, and context. Numerous environmental factors have been implicated in the etiology of MDD. Some of the most well-established ones are stress. Stress is a state of physical or mental tension that results from challenging or threatening situations. Stress can activate the hypothalamic pituitary adrenal (HPA) axis, which regulates the secretion of cortisol and other hormones that modulate various bodily functions. Chronic or severe stress can dysregulate the HPA axis and cause alterations in brain structure and function that may predispose one to MDD4. Stress can also trigger inflammatory responses that may contribute to neuronal damage and depressive symptoms. Trauma. Trauma is a type of stress that involves exposure to actual or threatened death, serious injury, or sexual violence. Trauma can induce psychological reactions such as fear, helplessness, horror, shame, guilt, anger, and dissociation. Trauma can also affect the development and functioning of brain regions involved in emotion regulation, memory formation, and threat detection, such as the amygdala, hippocampus, and prefrontal cortex. Trauma can increase the risk of developing post-traumatic stress disorder, PTSD, which often co-occurs with MDD7. Early life adversity. Early life adversity refers to negative experiences that occur during childhood or adolescence, such as abuse, neglect, poverty, malnutrition, or parental loss. Early life adversity can have lasting effects on brain development, neuroendocrine systems, immune function, and epigenetic regulation. Early life adversity can increase the vulnerability to stress and depression later in life. Social isolation. Social isolation refers to the lack of social contact or support from others. Social isolation can impair one's sense of belonging, self-esteem, and coping skills. Social isolation can also affect the activity of brain regions and neurotransmitters involved in social reward and motivation, such as the ventral striatum and dopamine templor. Social isolation can increase the risk of developing depression and suicidal behavior. Epigenetics' role in depression. Epigenetics is the study of how environmental factors can modify the expression or activity of genes without changing the DNA sequence. Epigenetic mechanisms include DNA methylation, histone modifications, and microRNAs. DNA methylation is the addition of methyl groups to certain cytosine bases in the DNA, which can affect the accessibility and transcription of genes. Histone modifications are the addition or removal of chemical groups to histone proteins, which can alter the structure and function of chromatin, the complex of DNA and histones that forms chromosomes. MicroRNAs are small non-coding RNAs that can bind to messenger RNAs and inhibit their translation into proteins. Epigenetic mechanisms can mediate the effects of environmental factors on gene expression and brain function, and thus influence the risk and course of MDD. For example, stress can induce changes in DNA methylation and histone acetylation in genes related to the HPA axis, neurotrophic factors, and inflammatory cytokines which may alter their expression and contribute to depression. Trauma can induce changes in DNA methylation and microRNA expression in genes involved in glucocorticoid signaling, synaptic plasticity, and neurogenesis, which may affect their function and lead to depression. Early life adversity can induce changes in DNA methylation and histone modifications in genes associated with stress response, emotion regulation, and cognitive function, which may persist throughout life and increase the susceptibility to depression. Epigenetic mechanisms can also provide a link between genetic and environmental factors in MDD. Some genetic variants may influence the epigenetic status of certain genes or regions, making them more or less responsive to environmental stimuli. Conversely, some environmental factors may interact with specific genetic variants to produce epigenetic changes that affect gene expression and function. For example, a genetic variant in the serotonin transporter gene, SLC6A4, has been shown to interact with stress exposure to modulate DNA methylation and expression of SLC6A4, which may affect serotonin levels and depression risk. In summary, MDD is a complex disorder that results from the interplay between genetic and environmental factors. Epigenetics is a promising field that can help us understand how these factors interact at the molecular level to influence gene expression and brain function, and ultimately affect the risk and course of MDD. Epigenetics may also offer new avenues for diagnosis, prevention, and treatment of MDD by identifying biomarkers, risk factors, protective factors, and potential targets for pharmacological or behavioral interventions. However, more research is needed to elucidate the specific epigenetic mechanisms involved in MDD, as well as their causal relationships with clinical outcomes. Furthermore, epigenetic studies should take into account individual differences in genetic background, environmental exposure, developmental stage, sex, ethnicity, and other factors that may modulate epigenetic processes. By integrating epigenetics with other disciplines such as genetics, neuroscience, psychology, and epidemiology, we may be able to gain a more comprehensive and holistic understanding of MDD and improve its management. Thank you for watching this video on what we know about the genetic predisposition for depression. I hope you found it helpful and informative. If you did, please give it a thumbs up and share it with someone who might benefit from it. And if you want to see more videos like this, don't forget to subscribe to The Anxious Resistance, where we explore various topics related to mental health and well-being. Your support means a lot to us and helps us spread awareness and education. Until next time, stay safe and take care of yourself.